Welcome to the Analysis Walkthrough RNA-Seq video, which is part of the 7 Bridges onboarding lesson for the Cancer Genomics Cloud. While watching this video, please remember 2 key concepts. The first is that the most basic analysis flow on the CGC involves project creation, adding files, staging apps, and finally executing analysis. The second is that on the Cancer Genomics Cloud, analysis outputs can be viewed and even subjected to tertiary analysis without having to download them. At the end of this video, you should be able to run a basic analysis that can be used as a template for your own work, run an app from the public apps gallery, view stats and logs, and ask for support directly from the task screen. I'm going to demonstrate running a differential expression analysis workflow starting from FASTQ files and ending with a list of differentially expressed transcripts. Let's get started. So as you can see, I'm logged into my profile on the Cancer Genomics Cloud. The first part of our analysis workflow is going to be creating a new project. I have a bulk RNA-Seq demo project here, and I'm going to be pulling files from that, but I want to create a brand new project. Call this RNA-Seq workflow, and I'll be using my pilot funds. Refer to the project creation video for all of these different settings. I'll hit create. And now I have this project. So now I've created this project and I'm going to need to add files to it. So if I go to my files pane, I can see that I have no files here. I'm going to add files from another project. So I'm going to click projects. I have this bulk RNA-Seq demo, and I'm just going to click on the input folder here. If I look in here, these are all the files that you should recognize from the file management with metadata and tags video. So I want all of them. I'll just click input and click copy to project. And then they all show up. So now I have my input files. I need to get files for references to align these FASTQ files to. So I'm going to actually get these from our public reference files. And I know the files I'm looking for, so I'm going to type in GCH38 ERCC. And I'm looking for this GTF file, which is annotations, and I'm looking for this transcriptome file. So I'm going to copy those to this project. I'm going to create a new folder, reference, and I'm also going to add a tag, reference. Now, when I look in that folder, there they are. So now I have my reference files. I need to get the apps that I'm going to be using for this workflow. So let's go to apps here, my apps tab. And we can see I don't have any apps currently in this project. I need to get them. So I'll click add apps, go to public apps. And for this workflow, I've decided to use a Callisto and Sleuth. So I'm going to start typing Callisto, and here is the Callisto Salmon Sleuth workflow that I want to use. I'll just click copy, and it says that the app has been copied to the project. And now, in order to use Callisto with my reference files, I also am going to need to make an index. So right down here, I have the Callisto index app. I'm going to copy that as well. App has been copied to project. So if I click this now, it'll take me to my apps, and I can see that I have these 2 workflows in my project. So I know that the first thing that I need to do is make an index for my reference file. So I'm going to use this Callisto index tool. When I click on this, it tells me things about the tool. I can also see what inputs I have to have here. I need the transcriptome reference, and I have the option of using an annotation. So to start this, I'm going to hit run, and that creates a draft task. And in this draft task, I have my ability to add my input files. So for my annotation file, I'm going to click select input files, and that will take me here to this .gtf. And then for my transcriptome FASTA, I'm going to again hit select files. And the apps tend to be pretty smart about what kinds of files are acceptable for any given input. That's part of the common workflow language wrapping. So as we can see, there's only a few different files that are file extensions that are acceptable here. And that's why I only have my transcriptome FASTA here to select. And so that has taken care of both of my required inputs. I also here want to create a transcript to gene annotation file. That's part of why I added my GTF file. So I'm going to click true. And that's everything that I need to do to set this up. And I will hit run. And that will go allocate the cloud resources and start creating the index file that I need to run Callisto. So while that's running, let's take a look at the differential expression analysis workflow we're going to be using. It's called Callisto/Salmon Sleuth workflow. We're actually going to be using Callisto and Sleuth. So when I click on this to go to the workflow page, the first thing I see is a wiring diagram that shows me how the different tools that are being used in this workflow are arranged, how the outputs of one tool become the inputs of another tool. And so I can see that for me to run this, all I need is the FASTQ files, which are going to be my in-reads, and an index file. And then out of that, I'm going to get an RData file that I can use for later analysis in RStudio, the list of differentially expressed genes, and an HTML report along with some other useful matrices. If I scroll down here, I get some further description of what this is. So we're using a workflow here that does pseudo-alignment. Both Callisto and Salmon do pseudo-alignment, or what's also called quasi-mapping, which lets us count the number of times a transcript is sequenced without having to do a full alignment. Pseudo-alignment is much faster than full alignment and is highly recommended if you have a well-described genome. The differential expression is actually going to be handled by Sleuth, which is an R package. And the thing that I think is very interesting about Sleuth is that it does bootstrapping. So we're actually going to give it a number of bootstrap replicates that we want it to perform. And it's going to use those many different iterations of the pseudo-alignment software to decouple biological variants from inferential variants. Let's scroll down all the way to the bottom. And we can see here, again, a list of what the inputs are and whether they're required or not. So we have to have the Callisto Salmon index file, and we have to have the input reads. We have an optional file called phenodata, which is a CSV file for phenotype data. If you have a multi-condition experimental design, this is where you would provide a CSV file that says which conditions apply to which samples. So in order to run this, we're just going to, again, click Run to start a draft task. So here's my draft task. And I have red fields here again saying which fields I must have an input for. I know that my Callisto index task has already completed, so I'll click Select Files here, and I see that I have a Callisto index now. I'll click that and hit Save Selection. And then I need my list of FASTQ files. Now, these are all in my input folder, but because the app knows that I'm looking for FQ, fastq.gz, etc. extensions, I actually can just see all 30 of these files here. And so I'm just going to click Select All with this drop down, and then click Save Selection. And so that's put all of my FASTQ files in. I am not going to use a phenotype data CSV. Instead, I'm going to be providing that information in the app itself. So now we have to do some app settings. So first we need to tell it which pseudo-aligner we're going to use. We're using Callisto. It's a touch faster than Salmon. And then we must tell it how many bootstrap samples we're going to use. I chose 100. We don't need to change any of the defaults here, and we're not going to use Salmon. We do not need to change any of the defaults here. And if you want, you can give yourself an analysis title. I'm going to do RNA-Seq demo. And now we have to, this is where we're actually going to tell Sleuth what our design matrix is, what the covariate of interest is. And in order to explain that a little bit better, I actually want to go look at the files and show you the metadata. So we'll simply scroll back up and go to the Files tab. And I'm going to restore default columns because I want to show you how you might go about doing this yourself. Click on Input. You see all of our FASTQ files. And to look at the metadata associated with these that's going to be useful to us in our workflow, go ahead and remove task ID because it's just going to take up space, remove created on, and then add genotype and add, it's called induced HBV replication. So these are the two conditions that our RNA-Seq samples differ across. We have some wild type samples and we have some KD genotype samples. And then some number of wild type samples are HBV neg, some number are HBV pos, and same with the KD genotype. So this is the metadata that I'm going to be using as covariate of interest, this induced HBV replication. So I want to make sure that I've got exactly this text. I'm actually going to click this file, open its metadata view, and I'm going to highlight and copy with Control-C that text because I don't want to make any typos with it. And now I'm going to go back to my tasks view. I'm going to click this draft task that we were working on. And I'm going to scroll down to where I was putting in the covariate of interest. And I'm going to hit Control-V to paste that induced HBV replication. Then the last things that I must select here, I want to do my DE analysis at the level of the gene. And I want to select human. And I don't have to do this, but I'm going to not skip the sleuth execution. That's also the default. This is now all set up, so I can simply hit run. And this task will be allocated a cloud instance and run. Once a workflow is running, you can actually view stats and logs to check on how the workflow is proceeding. This is also useful after workflow is completed. So from the task page, just click View Stats and Logs. And you'll see a timeline of what has been happening so far in this instance. We can click on this and it'll show us a little bit more information on the side about what task is happening. Here, the tool SVG Choose Pseudo-Aligner is running, and that's deciding which pseudo-aligner, Callisto or Salmon should run and setting things up for that to run. You can view instance metrics from this screen, and that will show you the tool's usage of the cloud instance that it's selected. So we can see the CPU is staying at right around 85%. We're not using disk writing very much at all. Our RAM usage is very good so far, though later on during Callisto, that RAM will spike back up. And we have our input-output metrics, a bunch of other metrics that are helpful, especially in troubleshooting perhaps why a task didn't run. So I can also show you some completed tasks from earlier to see what those logs would look like. Here in a completed task, we can see what the price was. This ran for 2 hours and 28 minutes and cost about $2. We can click View Stats and Logs and see all of the steps and how long they took. The Callisto Quant step, which ran on 15 different samples, and how those different Callisto runs went. And then this final sleuth step. Clicking on any of these will actually give us access to logs. So this means if you do have a problem, you can really kind of dig into what happened on the cloud compute. We have our error log, which is just the standard error stream that tells us about that sleuth run. Or we can look at the logs from Callisto Quant itself. We can see all these different Callisto runs and look at the error log. So what if we had an error? What if the task failed? This is what your output would look like in that case. You would have a red failed tag here and some error information here. This is telling me that something went wrong with my R script. You could go to View Stats and Logs, and you could click where sleuth failed, look at the logs, and try and understand from this what happened. In fact, it looks like the job was unable to get to the Ensemble service. It wasn't able to get to Ensemble over the internet. So now I think I understand what happened. Let's go to settings of this project and just see when this project was set up, block network access was selected, and for sleuth to be able to run, you have to allow network access. So that probably will fix that problem. But if you have a failed task and you're stuck, one of the easiest things to do is click this button, Get Support. Here you can just type a little bit of information to let our support team know what's happening. I tried to run Callisto and Sleuth and got an error. Please help. This checkbox, invite a Cancer Genomics Cloud support team member to this project, will give one of our support staff instant access to your project to see what's going on. And then you would just hit Submit Request. Finally, I just want to show you what the output of a successful completed task is. Here's one that I ran earlier with all the same settings. And what we see here is we have our output files available now. There's an expression matrix that's just named bulk RNA Callisto. If we click on that and look at the raw view, we can see that that is just a counts matrix, which would be very familiar to you if you've done differential expression analysis before. We also have an HTML report. And so the sleuth tool on the Cancer Genomics Cloud will produce this via our R Markdown. It tells you the run information. So here we can see that induced HBV replication was the covariate of interest that we named. It'll tell you that it's got 15 samples with counts over 892,710 transcripts. And then it does a very simple and quick PCA. I'm zoomed in a little bit too far to show you what this looks like. And it does it has a few other useful exploratory plots. At the very bottom here, it's going to tell you the top differentially expressed genes and their p-value. We also have an R data environment. So in a later video, I will show you how to start an RStudio session and load this R data into RStudio for further analysis. We have our normalized counts, which is, again, another expression matrix that you could use in RStudio and analyze this with DE-Seq 2 if you wanted to. And then we also have the sleuth analysis results, which is a CSV file. And this truly is the 4 differentially expressed genes and their p-values. So the key concepts from this video have been that the most basic analysis flow on the Cancer Genomics Cloud involves project creation, adding files, staging apps, and finally executing the analysis. We demonstrated this by running Callisto to make an index, and then running Callisto and Sleuth on RNA-Seq FASTQ files. The second concept is that the analysis outputs can be viewed without having to download them. You can view the output directly on the CGC. You can also run a Data Studio session for further exploratory analysis, which I'll show you in an upcoming video. That concludes the analysis walkthrough RNA-Seq video. The next video in the series is billing groups and pilot funding. Thanks for watching.